Good morning to everyone. I'm Kitley, Field Application Specialist in Apical Scientific. And welcome and thank you for participating in today's webinar with the title Routine PCR Life Saviour, DreamTaq DNA Polymerase. So when I go through the slide, okay, if you have any question, again, please write in the chat box, okay? So not to delay, let me start my sharing here. Okay, in this webinar, so I will start with a short introduction on PCR. Then we will talk about the function of DNA polymerase and tag DNA polymerase in PCR. Then after that, we will go into a more detail on the routine PCR application. And I will share with you how DreamTaq it can help you on your routine PCR work. And last, I will share with you some tips and tricks on work on your routine PCR. Okay. Okay, so we start with the short introduction. So what is PCR? I believe most of you know, but just a little bit on the PCR. So PCR is a polymerase chain reaction. So we start the words with polymerase, right? Okay, so PCR, it is a powerful tool used in life science to make copies of a specific segment of DNA. So it involves a series of temperature cycle that cause the DNA to replicate rapidly. So it allowing the amplification of a small amount of DNA into million of copy of the target DNA. And PCR, it has a countless application in the field such as like the most in molecular biology, okay, and also in genetics. It can be used also in forensic medicine, agriculture, food science. Okay, so it has many application to use in many different application. Okay. Okay, so how PCR start? A little bit on the history. Actually, PCR it start in 1970. So where the single-stranded DNA replication was reported, which it actually involved the use of primer and DNA polymerase. Then the tag polymerase was isolated from bacteria Thermus aquaticus, and this bacteria is actually live in high heat place. So the polymerase, it can withstand a high temperature that is needed to denature the DNA in PCR. So with that, the PCR technique was then invented by Carey B. Mullis, who determined his Nobel Prize at 1993. Okay, so from here, it actually start the very active of DNA PCR development, where the PCR machine was invented. Then later on, the first PCR kit was developed. Okay, so after that, the PCR become a so convenient and become a routine work that is widely used in many studies. Okay, so the PCR, we start the name with polymerase chain reaction, right? So it's a chain of reaction that using the DNA polymerase. So we know that DNA polymerase is the most important things in PCR. So in a PCR reaction, you will need all these components. So you will need the forward and reverse primer, and then you will need the DNA template. You will need the DNTP, which is the nucleotide block, okay, and also the reaction buffer and water, and certainly the DNA polymerase. Okay, so it is the most essential component that is among all these components. Okay, and DNA polymerase in the PCR reaction, it will help in synthesizing the new DNA strand. So understanding the characteristic of this enzyme is very important, which then it will subsequent um, uh, tell you how the DNA polymerase we can develop it into more advanced polymerase. Okay. So what is DNA polymerase? Generally, DNA polymerase is an enzyme that help create new DNA molecules by adding nucleotide to a growing DNA strand. Its main function include DNA synthesis, proofreading, and DNA replicate and repair. Okay, so DNA polymerase has two types of activity. The 5' to 3' polymerase activity, it allow it to read the original DNA strand and add nucleotide from 5' to 3' bases and form a complementary strand. Okay, and this activity is also important in DNA replication and also DNA repair, where it helps in copy DNA before the cell division and also fix the damaged DNA in our cell. Okay, so the other activity from DNA polymerase is called 3' to 5' exonuclease activity. So this activity, it allowed the DNA polymerase to check and correct the error in the newly synthesized DNA to prevent any mutation. And also this is called as proofreading activity. Okay, so I believe many of you hear proofreading activity before, but maybe you don't know. Proofreading activity is also called the 3' to 5' exonuclease activity. Okay, so all these function of DNA polymerase, they are crucial for genetic inheritance, cell replication, and also all the overall of the organism growth. Okay. In the meanwhile, with these two activity that is possessed by DNA polymerase, it is essential for the molecular biology application, especially in PCR and sequencing. The 5' to 3' polymerase activity, it helps in amplification, while the 3' to 5' exonuclease activity help in making sure the sequence that you determine in your PCR product is similar with the sequence from the template DNA. Okay. Okay. So um the most frequently used DNA polymerase is Taq DNA polymerase. So we need to understand a little bit on Taq DNA polymerase. It is actually a type of polymerase which commonly used in PCR, and it is originally isolated from a bacteria called Thermo aquaticus, which grow in hot environment like hot spring or the hydrothermal vent. Okay, and because of this, um, the Taq DNA polymerase, it can actually withstand a high temperature and remain stable and active even the temperature is high and around 95 degrees Celsius. Okay, so this makes it suitable for use in PCR, which involve cycles of heating, cooling, to melt the DNA strand apart and synthesize a new one. Okay, so Taq DNA polymerase, it can synthesize DNA quickly by adding around 1,000 nucleotide per minute. So this enable the rapid amplification of DNA and makes the PCR process faster. Okay, so that's why it is actually suitable for routine PCR. The production and purification of Taq DNA polymerase is relatively easy, and this make it less expensive compared to other polymerase. Okay, and the accessibility of this polymerase make it ideal for most of the PCR application. But there is also some features in Taq DNA polymerase that may not favor for PCR. One of it is the polymerase. It actually lack of proofreading activity. So it will result in higher error rate upon the PCR, and also at temperature below its optimum, which means at a temperature lower than 72 degrees Celsius, the Taq DNA polymerase can bind non-specifically to DNA, and it will lead to unspecific amplification. Okay. Also, the processivity of Taq DNA polymerase is relatively low, which make it easy to fall off from the DNA upon the amplification, and this has limit the PCR product size. Okay, and yeah, so the above is the features of native Taq DNA polymerase. Now most of the Taq DNA polymerase in the market, they have already been modified to in order to make it more suitable for different type of PCR. Okay, so I will share this on the next few slide. Okay, so the routine PCR. What is routine PCR? Routine means something that you do it daily, right? So routine PCR means the PCR that you will need to perform nearly every day, or maybe every week, or every week maybe have a few times. Okay, so routine PCR is typically performed by using tag or tag based blended DNA polymerase, so to amplify the specific DNA sequence, and it involves denaturation, annealing of the primer, and then extension by polymerase. So this leading to replication of the target DNA. So routine PCR is performed in nearly all molecular study to verify or to detect the presence of the gene, and also to fish out or get multiple copy of the target DNA region. Okay, so it is also used in various application like gene expression analysis, genotyping, or molecular diagnostic. Okay, so for a routine work, I believe all of you or all of us also want to have a PCR with minimum optimization needed, right? Certainly, we don't want to have too much of optimization. And for routine work, it should be a very convenient to handle, and also it will able to give us specific amplification, right? So I believe all of these are the what everyone wish for in their study when you perform your PCR, right? But in some cases, you may also want a higher yield or higher sensitivity for your PCR, so that the PCR product may be more suitable for some specific downstream application, like maybe sequencing. Okay, and also routine PCR, it should be able to um, at least amplify the whole gene fragment, so that is possible to be long, maybe like 5 kb. Okay, so the DNA polymerase that is used must carry some specific characteristic that is suitable for all these feature in routine PCR. Okay, so what is the characteristic? One of it is the processivity. Okay, the processivity of DNA polymerase is refer to the number of nucleotide added to a growing DNA strand in one single binding event. So it before the polymerase dissociate from the template strand. So different DNA polymerase, they have a different level of processivity. A high processive DNA polymerase, it tend to have higher affinity to their substrate. Okay, and then it can incorporate more nucleotides per binding event. So it will give you long PCR product and a more complete amplification. And DNA polymerase with high processivity also, it will spend more time to when it bond to the template. So they can actually complete more copies of the target sequence in each cycle. And this can result in a higher yield of the desired amplification product, which particularly useful when the template DNA is low in abundance. Okay, so another one, the characteristic of DNA polymerase, or I should say it's a advancement technology in the DNA polymerase. So it is the hot start technology. So um, traditionally, actually during the reaction setup of PCR, primer can bind unspecifically to the DNA template, or they may bind to each other, and this mispriming will can actually later on extended by the DNA polymerase, and it result in non-specific amplification in your PCR. So hot technology can help to solve this difficulty. So hot polymerase, the polymerase, it is actually a type of modified tag DNA polymerase. So it carry mixture of tag DNA polymerase together or bind with an inhibitor, which can be either an antibody or a body or chemical modification, or also it can be a aptamer. Okay, so all these inhibitor, it will bind to the polymerase and inhibit the activity when the temperature is below 45 degrees Celsius or at room temperature. So when the temperature goes up in a PCR, so when you put your PCR reaction into the thermocycler and you start the PCR, when the temperature go up, then it will actually release the polymerase, and so there will be amplification happen in your PCR. So this will also allow you to prepare your PCR mix in a room temperature, rather than you need to go and find ice and then do your PCR reaction on ice to prepare everything on ice. Okay, so this technology can help you on all this. Okay, and here show in the gel photo, so you can see that the non hot it can actually give some unspecific bands. So when you go with hot start, then it helps to give you specific amplification, and you can see that it when the amplification is specific, then it means that you will get more of your PCR product. Okay, yeah, so hot start will give you high specificity in your PCR. Okay, meanwhile, PCR is actually carried out in certainly in a buffer that will provide a suitable chemical environment for the activity of the DNA polymerase. So for an optimum activity of DNA polymerase, the reaction buffer, it need to have a pH maintained between 8 to 9.5, and this usually we will stabilize the pH by using tris HCl. Okay, and for tag DNA polymerase, the PCR buffer will need to carry a potassium ion at a concentration of about 50 millimole in order to promote the annealing of the primer. And with the potassium ion, it is actually contribute from the potassium chloride in the buffer. If the concentration of this ion is too high, it may inhibit the polymerase activity. Okay, and also polymerase also need to have the presence of magnesium ion in order to promote its activity. So usually the concentration of magnesium ion should be around 1.5 to 4.5 millimole in a PCR buffer. So depends on different type of polymerase. Okay, so if the magnesium ion is insufficient, it will cause failure of the reaction. But if it is excessive, so it may also affect the fidelity of the DNA polymerase, and it may cause the generation of unwanted product. Okay, so commercially, DNA polymerase, they often will come with the PCR buffer together. So this PCR buffer is usually optimized for robust enzyme activity, and therefore it is less optimization will be needed. And we highly recommend you to always use the provided buffer to achieve optimum PCR result for your routine PCR work. Okay, and also together in some PCR buffer, it may come with the loading die uh, or tracking die, which can actually give you conveniency when you perform, when you want to check your PCR product in agarose gel electrophoresis. Okay. Okay, so far we have understood the feature of the routine PCR and some necessary characteristic for DNA polymerase to achieve this feature. So now let's see how DreamTaq DNA polymerase it can help you in your routine PCR to achieve all the features that I mentioned just now. Okay. So this is a highlight for DreamTaq DNA polymerase. So it can give you a robust amplification with minimum optimization, and it is higher in sensitivity, and it can give you high yield of PCR product. It also can amplify long target up to 6 kb. Okay, so DreamTaq DNA polymerase, it comes with a hot start technology, so you can choose the DreamTaq with hot start technology also. And the PCR buffer, it can allow you to direct loading into the gel. Okay, so I will explain more and share the performance of DreamTaq in the next few slides. Okay, so unlike the native tag polymerase, okay, DreamTaq DNA polymerase is actually modified with enhanced processivity, which it can give you higher sensitivity and greater amplicon length and also yield. Okay, for example, in the gel show here, so you can see the comparison of PCR sensitivity and yield with DreamTaq DNA polymerase and also some commercial polymerase. And here it is test for amplification of product with size 956 base pair. So using human gDNA as template, and you can see DreamTaq is able to give high amplification, yield, and even the template amount is actually low as 30 picogram. Okay, and with good processivity also, DreamTaq can give you robust amplification with high yield for a wide range of target size. So as shown in the gel here, okay, so the amplification of target with different size ranging from 160 base pair to 7.5 kb, and you can see here DreamTaq polymerase and also a polymerase from vendor, and it show amplification for size more than 4 kb. Okay, but the rest is maybe less. Okay, but DreamTaq here, you can see that it can give you better yield and less unspecific amplification. So you will have less smearing rather than some unspecific band. Okay, even the size is big. Okay. Okay, so DreamTaq DNA polymerase, it come with own optimized PCR buffer, and every component in the buffer is well optimized to give you a superior amplification. The buffer, it come with pre-added of magnesium chloride, um, that which actually make it more convenience to use. Um, Unlike some of the commercial polymerase buffer, which you actually need to add in the magnesium chloride to the buffer by your own. Okay, and the well-optimized buffer, it allowed the amplification of a wide range of primer with different annealing temperature. So it means that there is not necessary to perform too much of optimization for your PCR. Example here, okay, for amplification of 613 base pair fragment, okay, the same primer set, okay, you can see that DreamTaq can give you good and consistent amplification, even the temperature yield is different. Okay, so this means that less optimization will be needed for your PCR, and you can even multiplex the PCR with different set of primer that they may have different in the melting temperature. Okay. Okay, DreamTaq DNA polymerase, as I mentioned just now, it has a hot start version, which it can help you to increase the specificity of your PCR. So by looking at the comparison here, we can say that all of the commercial polymerase has no problem to give specific amplification for short PCR product. Okay, so you can see all short PCR product, they are quite good. Okay, however, when the amplicon size become bigger, you can see that some of the polymerase will give you unspecific band. Even they are hot start polymerase. So this actually depends on what is the hot start technology used. Okay, but with the special modified DreamTaq hot start polymerase, the amplification here are highly specific. Even the amplification of big amplicon size like 5 kb here. Okay, so it can give you a good specific amplification with good yield. Okay. Okay, DreamTaq DNA polymerase can give you a lot of conveniency in your routine PCR. You can choose to use the green buffer format so that it is your handling for your daily work. So you can easily see your PCR buffer inside and PCR reaction mix inside your tube. Okay, the PCR buffer from DreamTaq, it carry loading die, so it allowed you to direct load your PCR product in gel for agarose gel electrophoresis. So it saves your time to mixing the product one by one with the loading die. I know most of us will cut a parfum and then we put loading die and we mix every time. Okay, so it it save your time in mixing all this. Okay, and the buffer is also carry the tracking die with the blue and yellow die, so it allowed the visual tracking when you run the gel electrophoresis. Okay. Okay, so all this is the family member of DreamTaq DNA polymerase. So you can choose based on your preference. Here you can see that there is the basic one. You have standard DreamTaq DNA polymerase or hot start DreamTaq polymerase. Okay, so both standard and hot start, you can choose standalone polymerase only, or a master mix which carry all the DNTP reaction buffer and also polymerase inside, or it can also be polymerase only with the green buffer, or the master mix with green buffer. Okay, so you can choose. Okay, but if you have difficulty to choose which to choose, okay, so can share with you here. This is our recommendation. So if you are doing standard PCR, just to verify the gene, or just to confirm is there any presence of the gene or not, then you can use standard DreamTaq DNA polymerase. So it is suitable for general application, and it is good to give consistent and high product yield. And also it able to generate long PCR product. Okay, so if you are working on PCR that may need higher specificity, especially if you study on genotyping, or maybe you perform multiplex PCR, nested PCR. Okay, so DreamTaq Hotstart, it is good to give specific amplification. And one more thing, you can set up the reaction at room temperature. Okay, yeah. Okay, so last title here. So I want to share with you all some tips and tricks that may be useful for your routine PCR. Okay, so here is the common question that you may have in your PCR. Okay, one of it is, how do I eliminate non-specific band in my PCR? I just mentioned just now. Yeah, so um, you can use the polymerase activity. Actually, prior the start of the PCR cycling, it can lead to non-specific band, and the easiest way is to use hot start DNA polymerase. Okay, so if hot start, after you use hot start, you still have um unspecific band, then you need to maybe increase the annealing temperature or decrease the annealing temperature. Okay, so to adjust a little bit on the annealing temperature. And also it is very important that you need to ensure your primer do not have cell complementary sequence at the 3' end especially. And also this the if the primer has self dimerization, it may also reduce the yield. Okay, so do make sure your primer is well designed. And maybe you can try to lower down the magnesium concentration. Okay, and also uh, last one is, you can try to perform a touchdown PCR to determine a optimum temperature for your PCR, for your primer set, and then you use the hot start to run your PCR. Or you can use nested PCR, which is a modified PCR method. Okay, so it designed to increase the PCR specificity. Okay, so another one is, if you experience smearing on gel electrophoresis after PCR, first eliminate the possibility of contaminant by using a negative of, or a no temperate control in your PCR to run together. Okay, and also ensure the proper casting of your agarose gel. So this you can actually check the when you run the DNA ladder. Okay, if your DNA ladder have smearing or no. Okay, then only you look into the PCR itself. Okay, very often and intense background smearing is due to overcycling. So this is caused by using too much of template, enzyme, primer, magnesium, iron, or the DNTP, or too many cycle that is used, or maybe also too low of the annealing temperature that is used. So to reduce the non-specific product, optimize the annealing temperature and extension time. And also it is important to ensure that your primer are well designed using primer tools, and it is not too old, means your primer, the best is less than 2 years, or try to get a um, better purity of primer. Okay. Okay, another common question that researcher usually will ask is, I I am getting no PCR product, or low PCR product yield, or actually there is no band at all for my PCR. So how? Okay, if this happen, you need to look into several PCR condition to optimize. Okay, so first, are there any PCR inhibitor that could inhibit the DNA polymerase? Maybe like a high concentration of um, hemine in blood sample, okay, or humic acid from soil sample. Okay, so if it it happened to be like this, maybe you can try to dilute your template, and then you use it to run the PCR again. Okay, or try to increase the amount of DNA polymerase, okay, or increase the PCR volume to dilute out the inhibitor. Okay, then look into optimizing the annealing temperature or magnesium chloride concentration. But the most easiest, okay, use DreamTaq, because the PCR buffer, you need lesser optimization. Okay. Also, you can optimize PCR protocol to ensure sufficient denaturation and elongation time. So also, one more important thing, do check your primer design. Make sure there is priming site in your template. Some of us, the primer design, it may be using the gene may be unknown, or de novo, then maybe you want to use nested PCR, okay, something like this to help you get your uh, gene of interest. Okay, yeah. So here come to the end of my sharing. Okay, thank you for participate in our webinar.